very happy to be here. Glad to be invited. Glad to be able to speak to you guys. What I'm going to talk about is what I work on at Rutgers University, which is really how plants use microbes to get the nutrients that they need. Also to do some other things. For example, to get stress tolerance. Microbes will benefit that. The other, plant stress tolerance, plant hardiness. The other thing is disease resistance. So bacteria in particular in plants are really part of the immune system of plants, the microbial immune system. And I'll talk a little bit about that. The other thing that I want to mention is that we have a little bit of time, more than an hour here. So if you have any questions as I go along, go ahead and raise your hand. And if something isn't clear, I'll try to make it clear. If something doesn't make sense, let me know. And I'll try to make it make sense.
I have the first question.
Yeah, you bet.
Would you share your slides with us?
Yes, you can. My slides, you're welcome to. James has them on his computer. He can give you the slide set, or you can email me,
Great.
And I will email the slide set to you. Yeah, so I have no problem. I view us, and I think you guys have the same mentality. We're all part of a team, right? Trying to change agriculture. And any information that I have that's on slide sets, you're welcome to have it. Actually, most of our publications in this topic, we put in open access papers so that they're free to be used by anybody. And I could send those. If you don't know where they are, I'll send those. Slide sets, whatever will be helpful to you guys, I would be happy to do. So this is a case, we've referred to this before, of plants, and you actually may have mentioned it. Plants don't need us, or it may have been someone else, might have been Wendy, I can't remember. Someone said it. Plants don't need us. They're able to grow perfectly on their own without us. No plant in nature needs any fertilizer or any agrochemicals. They don't. They do perfectly well without us. I would argue that the reason they do perfectly well without us is because they have microbes. And those microbes give them all the nutrients that they need. And I'll talk about this. I have this kind of a title, How Plants Farm Soil Microbes, because plants are farming those microbes like we farm the plants. And so let me see if I can get this to, okay, it's working. So plants are growing in soil with their roots into the soil. They're attracting microbes to those roots. They're internalizing those microbes into the root cells, and I'll show you the details of that. But essentially, these microbes go into the root tissues and cells, and they become what we call endophytes. Endophytes, and this actually is internal phyte, plant. These are really microbes that go into plant tissues without causing disease. And basically my entire academic career has been spent working on endophytes, what they are, and so that's about 45 years working on this area. And the rhizophagy cycle that I'm going to describe is newer, but it comes out of this work. And so you have some idea. Anyways, I have some special slides in here for you guys that relate directly to cannabis and hemp and members of that family. So this is my student, April Michi, who's working in this area. Very difficult to work in the area because it's not fully, it's still kind of regulated, so hard to work in. But we made some progress. Cannabis has endophytes in it, as you would expect. This actually shows it. If you take a cannabis, take hemp seeds, germinate those, and then you look at the roots, and this actually is a root hair of cannabis. No special treatment. We didn't do anything to it. Just germinated it and put a stain on it so that we could see the microbes. And this is the root hair. And you see these little spherical things in there? Those are all the bacteria that are naturally inside the cannabis root hairs. These are bacteria, they're all throughout the plant. We don't have to do anything to the plant to have these there. They're going to be there. These endophytes, they are fungi and they are bacteria, mostly bacteria. And they go into plant tissues, into the cells of the plant, and you would think they would cause disease, but they don't cause any disease. The plant basically has these microbes under plant control. Now, if you took the same microbe, you made a cut on a plant, and you injected the microbes into the cut, it might be different. But through the tissues that the plant uses these microbes for, puts them in, the plant is in control. There's no disease at all. So here's another endophyte. For anyone who's familiar with the, say, forage grasses, like fescue grass, there's a kind of endophyte there that people actually don't like. And it's a fungal endophyte, and you can see it here. This is inside the leaves of the grass or inside the stems of the grass, you get this little fungus. And these are growing between the cells. So this is where, this is actually this fungus called epichloi, but it's beneficial. It's a grass endophyte. Beneficial to the grass, but not so good for animals, because animals, they eat a bunch of this, it'll make them sick. But most endophytes, most of the bacterial endophytes and even other fungal endophytes in plants don't have any negative effects on animals. This is a rare one that does. And if we were to look at this plant in more detail, you'd see that it's loaded with a lot of other endophytes besides this one. In fact, a plant will take in from the soil a whole community of microbes. So they'll be full of microbes. And this is something different from what we normally think of as plants as doing this, but they will really become quite full of microbes. This is one of the beneficial things that I get to do is to go and do field work. And my field work typically is not like your field work that might be actually in the field. Instead, I go into different places, in this case, it was Bonaire in the Dutch Antilles, and did some field work. And this actually is a cactus that's present on that island. And these cacti have fruits at the top. Here you can see the fruits right there, like prickly pear fruits. And if you look inside that fruit, take the seeds out, you get something like that. Germinate that, you get a seedling like this. And you get the shoot of the cactus, that's the caduceus cactus, and you see the roots right there, and you can see the root hairs right in there. And this is stained now with hydrogen peroxide so that we can actually see bacteria inside the tissues. What happens is the plant will interact with the microbe using reactive oxygen. Superoxide, actually, and I'll talk about that. But because of that, we could take these reactive oxygen stains and then we can see the bacteria inside the plant tissues. Without this stain, these would be invisible. And this is the main reason why people didn't know about this process, is because there was no stains that they could put on the tissue to actually see them. Okay, this is the hair now. This is one of these root hairs from that caduceus cactus. And you can see now the orange color is actually this. These are all the bacteria that are inside that root hair. You can see just some of them are in chains like that. Some of them are independent, but these are all bacteria. They're living inside the root cells. They're alive in there. All plants have endophytes. Not a single plant doesn't. Now, in some of our cultivated plants, we've damaged the endophytic systems that they had, okay, but they still will have bacteria and fungi that go inside them. They just may not be the best microbes. We've lost some of the best ones because of the way we treat crops, because of the fertilizers we put on them and the agrochemicals and various other, the way we treat seeds, dry seeds, yeah.
So specifically in cannabis, I know in like traditional agriculture, we have chemical systems, but there's still, endophytes and microbes in those soils. What about like hydroponically grown cannabis seeds, where there's no microbes present for multiple generations? What type of effect would that have on its endophytic system?
So some microbes are definitely lost. There are microbes that survive in the system. I have a student working on hydroponic and aeroponic. Hydroponic is a little better than aeroponic. Hydroponic, when we put microbes on the plants can respond. In the aeroponic, the response is definitely muted. And we think the reason why is because the microbes are being washed off of roots by the spray, right? So they do lose microbes. There's no doubt about it. I can't, without specifically knowing a particular study, I can't say how bad it is. But yeah, I would expect that there are definitely some microbial loss.
But they can still maintain the system by creating a microbial system like on the outside of the roots.
You can put some back in, or you could treat them differently. What you have to do, there was some discussion before regarding fungi coming into systems. Either they're oomycetes like Pythium or Fusarium that can be a real problem. Typically what we see is that you have disease problems when you don't have enough bacteria there. And the actual solution, people typically will, to try to control their disease problems, people will use fungicides. But what we see is actually that's not the right response. The right response would be to put bacteria back in the system, and that would control the fungi. Okay, but yeah, they lose stuff, and when they lose stuff, that's when they are more susceptible to disease. Yeah. Okay, so let me see where I was now. I missed a few. Or is that back this way? It jumped ahead. I have to go back, sorry. Oh, I missed that. Look. This is good enough. Look here. What was it? One more back? Yeah. Okay, I'll get back. I'll go back, but I want to talk about this first. Leslie, I got to talk about this first, Leslie. Okay? Okay. When we cut into a fruit, we think it's sterile inside. You know how wrong that is? They are not sterile. They are, if you look inside this fruit, there's bacteria all in here. Around this tomato seed, if you go in the seed, if you look actually under the seed coat near the embryo, there's bacteria around the embryo. It's in there, there's a tissue called the nucellus that actually is the food for the embryo in the seed, and it already has bacteria in it. So this plant fortifies its seed with the microbes that it needs to survive. That's the natural system. That's the way plants naturally work. And so they're not sterile. And it became an issue before one time when I was saying this many years ago, 15 years ago. An extension person heard me at Rutgers saying that, and then that particular extension person told people it's not true. It was at the time when E. coli was getting into plants, and someone had mentioned, well, there's bacteria in there already, and so that extension person said, no, that's not true. They're not in there. Of course, that's important because to them, they didn't want people to be afraid that these plants were taking up poison, toxic E. coli, right? So I understand why, but it was wrong. The extension person was dead wrong. They're loaded with microbes. And This is one of the microbes. This is one called Micrococcus, but it's a nice microbe for us in the laboratory because it has a particular shape that's easy to track. And this particular bacterium produces these tetrads, clusters of 4, as part of its cell. And so we can put this in a plant and we can see where it goes in because it has those tetrads. And let me see if I get that wrong. Okay, yeah. So that's an endophyte in tomato. Now this is a tomato seedling root. And what we see here is at the very tip of the root, this is the root cap cell. You can see that typical root cap cell. Those cells fall off as it goes through the soil. Then behind that we have the meristem. This is where the cells are dividing. The cell walls of these cells are thin here. They're soft as soon as they divide. They're really soft. Bacteria actually enter the cells in the root, right in this zone. And what happens is these cells have thin walls, and the plant also calls these microbes from the soil by secreting exudates. And you've probably heard that plants will secrete as much as 40% of their photosynthesis sugars as exudates. They'll just secrete them out here. Well, those exudates are actually signal molecules. They're attractants. It's bait for the soil microbes. The soil microbes will detect the exudates that are being secreted here, and they'll come swimming. And then the plant will cultivate them right in here. And then the plant will take them into the root cells here, to the actual cells. And I'll show you that. Then once they go in, the plant in this zone, just behind that tip, the plant will hit them with superoxide. Superoxide is a very potent form of reactive oxygen, and that'll just extract nutrients from the bacteria, but it also will take the cell walls off of the bacteria. And a lot of those nutrients come in those cell walls. It'll just oxidize the cell walls off. So this orange zone, that's where that's happening. This orange, by the way, color is ethylene, because the microbes are secreting ethylene. And that's how the plant knows the microbes are there. That ethylene is actually a growth hormone that these bacteria are producing. The other thing you should know is this actually is in this zone of cell elongation, it's called, in the root tip, right? And then if you look past this, so this zone of cell elongation is also the zone of nutrient extraction, right in here, nutrient extraction zone. And I'll show you how that works. But you can see older part of the root, now you see root hairs forming. The root hairs form here. When those root hairs form, in fact, the root hairs don't form unless there's bacteria in those hairs. And when these hairs start to form, these bacteria will be ejected from the tips of these hairs back into the soil. I'll show you that process. I'll show a film of that so you see that ejection. But these hairs don't grow unless there's bacteria in the root hairs. So the plant can't even develop without bacteria in the cells. So they are so dependent on bacteria, soil bacteria, that they can't even develop properly. There's no root hairs. Plants also roots don't branch as much. There's all kinds of, they won't grow nearly as well without those bacteria. Okay, so this is actually a part of a root or near the meristem tip of a plant called curlydock, which you're all familiar with. Curlydock, Rumex crispus, Rumex barnyard dock, named a number of things, but we can clean the bacteria off of it, the seeds, and then germinate them and put our bacterium on. And that's what we did here. We cleaned off the native bacteria off of this curlydock. We put our Micrococcus, the tetrad forming one in. And the whole thing I wanted you to see this is because you can see these are the root cells right around that meristem area, and you can see the tetrads of that Micrococcus inside. So you see they're inside the cell. And all these microbes are soil microbes. They all come from the soil. Every microbe that I'm going to talk about is from the soil. 99% are soil microbes. But what the plant will do the plant will bring them into the root, and then we'll train them to grow inside the plant. And we'll select the ones that are really good. Say it's a good nitrogen fixer, for example. And then they'll move that bacterium from the root and they'll move it up to the seed. And then they'll put that in the seed. Those are the ones that'll be on the seed. Those are the best that plant has acquired from the soil. So they're taking a lot of microbes in the soil, but the best they're putting up on the seeds. And that, of course, is a reason why we need to be careful with seeds. If you sterilize seeds, it's not a good idea. You kill those microbes. If you store the seed under high temperature conditions, for example, those microbes will die in there. And when the microbes will die, your germination rate will go down. There's an endophyte person, a scientist in Maryland, who actually did a patent for putting microbes, endophytes onto plants, a bacterium back onto plants, and get the germination rates to go back up again. So he actually did a patent for that phenomenon. But that's one thing that happens when the germination rates go down. At least some of the time, it's because the endophytes have died in those seeds. So we could reacquire them. In 2010, a group in Queensland, Australia, discovered that plants were internalizing microbes into their roots. And these are actually a couple of these investigators. Channy Pongfu was one. The other one, Suzanne Schmidt. They haven't really worked on this since this work, but they get the credit for this discovery. And what they found is that bacteria and yeasts are going into root cells. And so this actually here, this is a figure from their paper. This shows a root hair. You can see these little yellow dots in there. Those are the yeast that were internalized. These are smaller cells. Those are bacteria. That's in root hairs, actually. Shows the bacteria there inside. And the bacteria they used was E. coli, laboratory strain that they had. And what they did is they labeled them with green fluorescent protein so they can give it a tag, right? So it's going to be permanent on the microbe. Then they feed it to the plant, tomato and Arabidopsis, and then they look at that with fluorescent microscopy to see where the microbes are there. And you can see they're in there. They called this rhizophagy. Rhizo for root, phagy for eating. And this is the idea that plants are eating microbes in their roots. That was their concept. And we started to work on this right about the same time that they made this discovery. And we were actually working on it a little bit before from a different aspect. But after we figured out that the bacteria are actually inside, then we started to look inside the root cells. So the barrier, even for us and for everybody else, was that these microbes were going into the cells, and everybody knows they're not supposed to be there. So people, if they did have an inkling there's something there, well, it can't be. If they thought they saw something, well, it can't be. It can't be. This is the case. People just didn't see it. We could go to old papers, and we can look in some old articles, and we can see they have pictures of root hairs, and we can actually see the bacteria in the root hairs, and I'll show you some of that. And we could see some of the bacteria coming out, right? But the authors of the papers never described it because, of course, they're not supposed to be there. They weren't supposed to be there. That's where our concepts kind of get in the way of understanding new things. Well, what we found is that this is actually a cyclic process, and that what happens here is you get your root diagram of the root here, got the meristem here. The microbes in the soil are being attracted to that root tip. They're being internalized. Nutrients are being extracted using superoxide to essentially degrade some of the microbes and take their cell walls and take their nutrients out. The surviving microbes go into these little root hair initials, and then those microbes secrete growth hormone that causes these hairs to elongate, and they'll elongate in spurts. And every growth spurt, they eject some of the bacteria out, like that, all the way. And I'll show you an image of that so you can see how that goes on. If we remove the microbes, we get no hair formation. The microbes have to be there. It's a requirement. You cannot grow plants normally without microbes. So we termed this the Rhizophagy cycle because of this cyclic. It's a cyclic process. And that was a paper in 2018. This is a little cartoon of this done by an Austrian scientist, and you can see her here. This is Dr. Harriet Mella, and that's her with her daughter. But this shows Rhizophagy cycle and the way it works. So this is a root. You can see a microbe coming in, attracted to it, goes in now, nutrients go out of it. It goes into the hairs, triggers root hair elongation. You can see it there again. This is accurate in the sense that most of the nutrients come out of these microbes right behind that meristem in that zone. But some of those microbes will go into the root hairs and they start fixing nitrogen. And I'll show you that. Root hair, the root hair length of the root hair, all the microbial, all the processing of microbes in those hairs has to do with nitrogen fixation. And that's something we didn't really understand until very recently. Rhizophagy cycle happens at root tips. So if you consider, this is a corn, a diagram, a very old diagram from 1927, a botanist named John Weaver. It's old, but he illustrated very carefully this corn root system. And you can see all these little branches. All those branches are branch roots. And at every tip, that's where Rhizophagy cycle is happening. So there are thousands and thousands of root tips where Rhizophagy cycle is happening. And the plant is getting nutrients at every single root tip like that. Okay, this shows a branch root. This is in one of the test plants that we use. This is one called a common reed grass. But you see the branch root here. The meristem is right around here. You see this is all bacteria here. All this blue, you can see where it's dense here and it gets lower concentration out here. This is where the exudates are. This is the microbe cultivating those bacteria now from the soil. And you can see them out here as well, calling those microbes to the root tip to internalize them into the root. This actually shows what's happening. This is a root tip stained with a stain called crystal violet for the cell walls of bacteria. It'll stain these cell walls dark blue. And what you see is this is the zone where near the tip where the bacteria go in. You can see they're kind of rounded here. Those little dots there, those are bacteria. Rounded here, behind that zone, this is actually where the nutrients are extracted, where they're hit with superoxide. You can see these angular, that's cell walls coming off of these bacteria. They've lost their cell walls. They form these protoplasts. Those protoplasts, naked protoplasts, they're just bacteria without cell walls. And the plant gets nutrients off of these cell walls. That's the biggest part of the nutrients that they acquire. The non-nitrogen nutrients that they acquire come off of those cell walls, we think. Okay. This is a close-up of that. You can see the root cells here, and you can see the dark. That's all the cell walls that come off. There's a little bit of pink in there. Those are actually the bacterial cells there. So they're losing their cell walls as they divide. This is what it looks like after the cell walls are gone. This is a root cell, and you can see it. You can see it right here. This is the epidermis of the root, and you can see these little dots in there. Those are all the bacteria there, little clusters. And what's notable here, you can see there's a chain over here. You can see the smallest one, which is the youngest one, is staining blue. That's got protein in it. The blue, that's the blue stain. Aniline blue is what we use there. These bigger ones, these older ones, are not staining blue. They're losing their protein. That superoxide will go all the way through the membranes and clip the proteins up. And some of those cells will just break down and die right in there. So some bacteria, so we could put the picture together, some bacteria are going in and they're being degraded fully. Some of them go in and they're partially degraded. Some of them go in, if we force them to go in, and they're not broken down, and they're a problem for the plant because they're too oxidatively resistant. But the plant is taking in the ones that it can work with. It's not taking a lot of these in that it can't work with. We can force them to go in, but anyways, you can see that degradation in that cell. But these keep dividing, and so you keep getting young cells in there. Okay, we did a similar experiment here. My graduate student, Celeste Zhang, took a bacterium, labeled it with this tag called M. cherry that we could see it under the confocal fluorescent microscope. And then she scanned now, she's looking at these root cap cells that came off, and you can see the blue, that's the wall, and you see the cherry in there. Those are all the bacteria that were labeled with that. So they're inside. You know, all throughout the root of the plant, we track these microbes. Where these microbes are actually going in the root cell is, okay, this is a diagram of a root cell. This gray is the cell wall. They actually go just underneath that cell wall. They pass the cell wall, go in. How they get in, we don't exactly know. We can see them going in. We can actually look at them with the microscope, and we can see them passing into the cell, passing into this periplasmic space. So this is the cytoplasm here. So they go in underneath the cell wall, and then they really become functional in this little space between the cell wall and the plasma membrane or the cytoplasm of the cell. So they're functional in this. This is called the periplasm, or the periplasmic space. And it's in this space that the plant puts superoxide, this 2- superoxide that the plant is putting to degrade these microbes or extract nutrients from the microbes. The other thing that's happening, and I'll show you this, the plant is moving these microbes around and around, around like that. We think that does two things. That'll break up these bacteria into units, to smaller units, and force them to fix nitrogen from the air so that they can then get the nitrogen out of the plant. That's one thing that's happening. The other thing is that because the plant is moving them around this periphery like this, it keeps these bacteria off their toes, so to speak. If the movement stops, the bacteria can produce enzymes that they can degrade their way out. But if the plant is moving them, they can't get out. They're kind of off balance. The plant keeps them going. They can't get out. They can't escape. The plant is in control. That's the point. The plant is in total control of these microbes. The plant produces superoxide and puts on the microbes to get nutrients out of them. Superoxide is a highly potent form of reactive oxygen. It is short-lived in that it doesn't last very long and it doesn't go very far, but it's very potent. And the enzyme that the plant uses is this one called NADPH oxidase, or just NOx for short, NOx enzyme. And this is in the plasma membrane of the bacteria, of the root cell, of the plant cell. So this is an enzyme there, and it's this enzyme that makes that superoxide to degrade or extract nutrients from the bacteria. One thing that's notable about this enzyme is it has to have oxygen. And it takes that oxygen from the air, and it produces superoxide out of that. And that's what it uses to get nutrients from the bacterial cells. But this oxygen, if the plant, for example, if you have an area of the field that has poor aeration or flooding there, and there's not good oxygenation there, then plants can't make superoxide and they can't get nutrients from the bacteria. And so what you'll see in those soils, in those areas, is you'll see plants not growing very well. And they may also get diseased. And part of it is because they simply cannot get nutrients from these bacteria that they take in. The other thing is this. The cofactors are iron. And so plants need iron in order to do rhizophagy cycle. And if you're using something like, for example, an herbicide that is a chelator that will take specifically mostly iron, right? You take that, chelate that, and the plant can't get it, then the plant cannot do rhizophagy cycle. It can't make superoxide. And what that means is if you've got a crop, for example, that may be, herbicide ready, and you're putting the herbicide on it, you may in fact be addicting that plant to fertilizer because that plant can't get any nutrients directly out of microbes. So you're stuck squirting fertilizer, liquid fertilizer on those plants. So have to be careful with that. Oops. So anyways, this is what the bacteria look like with their cell walls. They may be rods like this or whatever shape. Once the superoxide oxidizes off those cell walls, they look like this, just little budding cells. This is long known. This is I think in the 40s or 50s, I think, they discovered these kind of protoplasts at the Lister Institute. And so they called these protoplast phases, they called them L forms. L referring to Lister Institute. This has been known in medicine for a long time. Back in the 70s, this is also studied quite a bit in plant pathology. But without microbes, I've already said this, you get no root hair elongation. The other thing that happens if you remove all the microbes, plants don't know which way is down. Roots, for example, if you have no microbes there, roots will go in the air, or they'll go flat on the surface. Very rarely do they go down. You put the microbes there, and then suddenly they know which way is down. Why that is, we don't exactly know. We do know that we can get different microbes that will, for example, make the root go down, and then some other microbes that will make the root get root hairs. So apparently, different microbes are producing different hormones that cause that. Okay, so this is an experiment where we took a Bermuda grass seed, we sterilized it, we germinated it, and we sterilized it to remove all the microbes on it. To do that, we actually had to sterilize it for like an hour with Clorox for a long time. More than most people do for seed sterilization. They'll do maybe 5 minutes at 10% Clorox, something like that, but you really have to sterilize it to get all the microbes. So that's what we did. Sterilized it really well, and you can see here, this is the older part of the root. You can see these little bumps. Those are the root hair initials. So roots will have these little pockets on them, root cells. Okay, but without bacteria there, they don't elongate. They won't grow. Have to have those bacteria there. This is a root now, Bermuda grass, where we put the bacteria on the seed. We remove them, then we put it on, and you can see now we got hairs immediately. All those hairs forming. This is older part of the root. And you can see the hairs out here. And you see the little brown dots. Those are all bacteria in there. They're full of bacteria. And even on these cells here that haven't elongated, those little dots there, you don't have hairs there, but you got little dots there. Those are all bacteria inside those cells. You can see some bacteria on the outside, but you see it's not staining brown like that because there's no hydrogen peroxide on the outside. It's only on the inside, because that's where the plant is secreting the reactive oxygen. This is a root hair now, one of these root hairs. You can see all these bacteria in these root hairs. You can see all those little brown dots. So they become loaded with bacteria. They're fat with bacteria. They don't just every now and then a bacterium goes in. They're eating these bacteria. They're engorging themselves on bacteria. These are really important. Yeah, Leslie?
Just a quick question. Can root hairs become roots?
No, the root hair itself will not grow into a root. The branch roots will grow from the bigger roots from inside. Okay, so these hairs are only superficial. They are extensions of the epidermis. And as it turns out, this is where most of the bacteria are going into these epidermal, the 1 or 2 layers on the surface. So this is where the bacteria are going. We used to think that the root hairs, the only function or the main function of the root hair was to extend the surface area of the root so they can absorb more nutrients, right? That's the conventional wisdom. But it looks like, from the work that I'm describing here, that those root hairs actually function as elevators to move those bacteria back out into the rhizosphere, right back into the soil, away from the root, right? It doesn't do any good just to drop them right at the root surface, but move them out away from the root and then put them out into the soil where they can acquire more nutrients. So the function of the root hair may not be what we thought. So here's tomato. Okay, tomato. We remove microbes, streptomycin, antibiotic treated. You see the root tip, no hairs. Here's where we did not treat it with antibiotic, and you see root hairs. Root hairs forming right away. Without those bacteria, you get no. You get, this actually shows this. If you look here, this is what the initial looks like, that root hair initial, but there's no bacteria there. That doesn't elongate. You look at these with bacteria, these little brown dots, those are the bacteria. These elongate. You can see the bacteria pressed out at the end. Here's a bit longer root hair, and you can see more bacteria there. Actually, this doesn't give it justice. There's so many more bacteria that are produced in these hairs over time as these hairs elongate. And I'll show you that, so you see kind of what I'm talking about. Why is this linkage between presence of the bacteria in the hairs and growth of those hairs, or presence of bacteria in root cells and growth of those root cells? And the reason is because this is my student who worked on this, by the way. This is Ivy Chang. Here you see a bluegrass without bacteria. You see there's no hairs. Here's with bacteria. You can see the hairs. And you can see these smaller ones. You see the little round dots there at the tip? Those are the bacteria that came out of those growing hairs. We look closer at them, I'll show you that, but they're bacteria. Why is there this association, this growth? I already mentioned it, because their microbes are producing hormones. Hormone, one of them is ethylene. That seems to be the main one. The other one is nitric oxide, or forms of nitrogen that actually happen inside those root hairs, and I'll show you that. Okay, here's a little sedge, and it grows on these limestone coral formations, but you can see the sedge here. There's no soil here. There's sand, but there's lots of microbes. Lots of microbes there. Okay, this is what one of those sedge hairs look like. And you can see the bacteria here around the edges. There's several of them in there. You can see the brown, but the blue in there is where the bacteria are. This is from the reverse of the Petri dish, so you can see it the way this hair actually works. This is how a living hair works. Hairs don't just sit there like we see here. They actually are in constant motion, constant movement. You can't see that if you pull it up from the soil and then put it under a microscope. You won't see that, because what happens is this motion, this cyclosis, is due to the cytoskeleton inside this hair. And if you move it, you break the cytoskeleton. And it'd be like taking a person and running over with one of those big flattener things and crush all our bones. We can't move. Well, that's what happens with these. You pull them up, it moves them, they don't work. But this is what they actually look like. They're moving these, so you can see these clouds. Let's see if I see them. There's some. Those are bacteria going around here. They're processing these bacteria. They're cycling these bacteria. This is really how the hairs work. This is one of the things that we didn't see, that people haven't looked at, because every time we pull it out, it stops. But you can see here, this is a hair. Now you can see clusters of bacteria here in there. And you see some are big, clear, some are little and dark. These probably all came from a single bacterium. So what happens is bacteria go in, the plant gets nutrients out of them, and then replicates, clones the ones that are left inside, and these are the ones that will be put back out into the soil. And I keep hitting the wrong button. This is what this looks like. Now you can see this is one of these root hairs. You see the bacteria going around, and you see them accumulating right there in the tip. They're bumbling around right there in the tip, and in mass they secrete ethylene and they secrete nitric oxide, and that causes a growth spurt in this root hair. And when that growth spurt happens, these bacteria are pushed out of the tips of those hairs. This shows one of these hairs again. And you can see this is a sedge hair. This is really fat. You can see the bacteria in here, but also all this white, that's all bacteria. They're just not staining with that stain because they're not touching the membrane. They're not touching the root hair is a little skinny thing in the middle of this. So all of these are around that actual root hair. And you can see out of the tip, you can see these bacteria coming out. And they're reforming. Once they come out, they reform their cell walls. They form rods back out here. And in fact, to let you know the extent to which the plant is in control of this, the plant puts a little bit of nutrient, little exudate that comes out with these bacteria. And that enables the bacteria to reform their cell walls and reform their flagella and then go swimming back out into the soil. So the plant is feeding these bacteria even after it extracts nutrients from them. And we can see that. But this actually is a growth spurt process. And this actually will a little diagram that shows that. And what happens is the bacteria will accumulate in the tip, and then they will produce their hormone, and then that will cause a growth spurt, and they'll be ejected. A few will be remaining here on the sides, around the edges. They will then be replicated again. They're going to processed again with cyclosis, increased, and then as that builds up again in the tip, then they'll produce hormone again, and then another growth spurt. Boom, 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 like that.
What's in it for the bacteria?
Say it again?
What's in it for the bacteria? What do they get out of this?
You know, a few go in and many go out. So I assume the bacteria benefit tremendously because the plant is cultivating them and putting them back out in the soil. What does a cow benefit? Because we take care of it and reproduce them and feed them. We eat them eventually. Well, that's what happens here. So this is like agriculture. This is like, a symbiosis like an agriculture symbiosis. Yes?
So essentially the plants are agriculturally cultivating their own food source.
They are.
They're farmers.
They're farmers. Plants are farming. Yeah. Plants are farming. Plants have agency. Plants are capable. They don't need us. We know that already. You guys know that already. Yeah.
So plants, basically have been around for 200 million years, and microbes have been around a lot longer. Could it be argued that microbes evolved plants to feed them?
Maybe. We have some, the earliest eukaryotes were, they weren't eukaryotes, right? They're archaeans, right? They came from archaeans, and through invasion of prokaryotes, right? Prokaryotes, blue-green algae for chloroplasts and mitochondria, purple sulfur bacterium, right? More than likely, eukaryotes have been doing this from the earliest, from the very beginning. They're internalizing these other microbes, right? That's why we have why we're so complicated. We have all this stuff because we assume we have absorbed them. If you look at the genes, at plant genes, we have genes of microbes, or the plants have genes of microbes. Animals, too, have genes of microbes, different species, all in our DNA. Yeah.
So that thought process, fungi is a microbe, but it's also a plant, right? Because bars see fungi everywhere.
Yeah.
So it's a microbe, but it's a plant. So chicken or the egg.
It is a microbe, and it can be internalized into the plant, and it obviously is a little bit different. They are internalized, but I don't.
But it's just terms that we understand.
There are terms that we understand. There is another process, and I'm not really going to. Well, I guess I'll show this. Some of the fungi are also taking in bacteria. Also, they absorb them into their tissues in the soil. You'd think they were living with them, pretty close to them. So they are also internalizing some of these, some groups more than other groups. It's phenomenal. It's an interesting phenomenon, and we don't yet understand that. I'll just allude to it a little bit.
So do some bacteria take in other bacteria?
I don't know. We predict that there are some archaeans, the so-called Loki archaeans that they think may have given rise to eukaryotes, the earliest eukaryotes, right? We think maybe some of them are really big cells. We think maybe they might have been doing this, but we're not sure. Yeah, because it's hard to study those things. But yeah, there are some, they're not really bacteria though, the archaea, but they're bacterial-like, they're prokaryotes. Yes, we used to call them all bacteria. But yeah, we think that there's a lot unknown, there's more unknown than is known about this. So, I mean, I'm just here to tell you we don't know everything about this yet. Yeah.
The endophytes that are actually surviving and then getting shot back out, in this signaling, do you think, or is there any evidence that they're actually being programmed to go mine a specific element to bring back to the plant so that it can become a host again and get let back in?
It would be fantastic if that were the case. Obviously, it could be the case, right? What we think instead, though, is that the plants actually will control what's in their exudates. And they will put certain exudates out. If they need, for example, nitrogen, what they will do is they will put the sugars out and they'll put the organic acids out in those exudates, but they'll withhold nitrogen. And then microbes that can fix nitrogen are the ones that can survive in this, follow that exudate stream, can survive at the tip, right? Because they have to get their own nitrogen. So we know that with nitrogen, there is that mechanism that they can control their exudates and attract certain ones. And we know that works. That's what happens, is the ones that fix nitrogen come. So with other nutrients, there are also other peculiarities of how the plants will change their exudates to favor other microbes. So the plants are not telling the microbes what to do, but they're selecting the microbes, right? They're selecting a whole community of microbes. It's not just one or two. It's not like, not when we do a biostimulant, we'll have a biostimulant, we'll have 3 or 4 together and we'll make a consortium based on what we want to do in there, what we think is necessary, what we think is good. But the plant is taking in a whole bunch of soil microbes. So it's different. The plant is doing it very differently than the way we would feed them microbes. They take in a lot, big community of microbes. So, okay, let's see okay, this is good. So this actually shows that is a root hair. And what we did is we took a bacterium out of another plant, not this particular, this is a root hair from the Bermuda grass. We took the bacterium out of a dicot, and then we put it on that plant. We removed all the other bacteria, we put this one on, and it worked. It went in, but with a peculiarity. It didn't get fully ejected. And still, what you can see is, you can see the dark area here, dark area here, dark area, dark, almost like it's banded, dark and dark. Those are bacteria that got stuck inside the hair, didn't get ejected. And so the clear area is where we had the growth spurt, and then we had a more accumulation of bacteria. Then there was an attempt to eject the growth spurt. They didn't eject, but instead you have that growth spurt, and then you have more accumulating. You see the point? Like that. Every 30 minutes, every hour, they did a growth spurt, and you see the bacteria there. A few bacteria went in, and a lot of them get replicated and go out. So the plant is populating its soil with these microbes that work with. This is tomato. This is my student that did this, but she made a movie of it. And you can see the root hair here. And you can see, if you look closely at this one, you'll see like 3 little holes right there, 3 little pores. And those bacteria are just slipping right through those pores. If these bacteria had reformed their cell walls inside, it would be plugged. So it has to be right? It has to be a match. So there's a match, and so it goes out. Now these would reform their cell walls and then go out and acquire more nutrients. We've done lots of experiments with plants with and without microbes, and we always get many more nutrients when we have the microbes there than when we don't. But the question is, which of those nutrients are oxidatively extracted and which ones may be solubilized, right? For this process, it's oxidative. So how do we measure that? Well, what we figured out that we could do, one approach, is we could inhibit superoxide in the plant, right? Make it so the plant can't make superoxide. So we can do that with elevated carbon dioxide. So we can put CO2 in the air at very high concentration, saturated CO2 environment, and then see what nutrients the plant can absorb in those circumstances. And this actually shows where we did that. This is a wheat root tip, and this is stained for superoxide. All this purple there, this is in the air, right, with oxygen, regular air, with just a normal amount of CO2. Then in the elevated CO2 environment, here you got the root tip again, and now you see it's also stained for superoxide, but you see no purple out here. In the middle, yeah, there's something in there, but the CO2 didn't reach into there, but it did inhibit the superoxide right in this zone, right in the outside. So then we measured the nutrients, and here's just one example: manganese. Right, this is in the elevated. This is the amount of manganese that went into plants in the elevated carbon dioxide environment. Okay, and so this is the one where it was in the air, and you see there's a huge difference between in the air, where you have oxidative extraction, versus where it has to be just solubilized. You know, what's coming from the soil water. It's not oxidative. Okay, and as it turns out, for manganese, this difference is like 136% to over 700% increase. A lot of the divalent metals are like this. They seem really important for this process. But of course, nitrogen and phosphorus and a lot of other nutrients are also important. Okay, but these divalent metals seem especially important, and they really are important for plant growth. So you would think plants would have a special, if there was limiting, they might develop a special system for getting nutrients out of them. And so Rhizophagy cycle, and I should say the earliest plants that had roots are doing Rhizophagy cycle. So as soon as roots came about, we see this process happening. Things like ferns, for example, that has nice root systems will have roots, Rhizophagy cycle, microbes in the roots, and so forth. So as soon as plants evolved roots, they were doing this process, Rhizophagy cycle. Okay, well, there also is another process, and that is nitrogen fixation. I alluded to it. The earliest plants on Earth were processing microbes in their tissues, in particular in certain hairs, filaments that they would produce. And for example, this is the team that's working on this, but this is bryophyte. These are seedless plants. These don't even have true roots. Okay, they don't make seeds, they don't make true roots. They're just really, really simple, little mosses. Okay, and you can see we got leaves here. That's pretty standard. They got chloroplasts in there, but you see these little brown filaments, those little threads. Those are these filaments I'm talking about. Those are where they make nitrogen in these plants. And if we look close at that, here you can see them on these little moss stems. You can see one of these filaments, and you can see the bacteria there. This is stained for nitrate. So this blue stain is nitrogen around those bacteria. Oops. This is actually a liverwort. Another very primitive plant, no roots, no root system, no seeds. But this thing, if you look at this leaves, you can see this is what they look like. You can see they have chlorophyll in them, chloroplast. That's pretty standard, right in there. And then you see the filament right there. There's no chloroplast. And you can see little blue dots. Each one of those little blue dots has a bacterium in the middle of it. That's full of bacteria. And they're fixing nitrogen, and the plant is getting that. Now, what's notable is you don't see that purple here where you have chloroplasts. And the reason is because when you have photosynthesis, they're making oxygen. And oxygen inhibits nitrogen fixation. It inhibits the nitrogenase enzyme. And so if there were chloroplasts in here, you wouldn't get any nitrate. You wouldn't get any purple in here. They wouldn't be fixing nitrogen. They could not fix nitrogen. They're incompatible. So plants had to develop these filamentous structures in order to do nitrogen fixation in them. We can see bacteria actually inside these cells, inside the cells here, but you don't get nitrogen. They're just in there. They're all in the plant, but you don't get nitrogen. It's not functional, at least not in that sense. This is actually a fern. We're in Massachusetts, so it's the Boston fern. And this is the gametophyte of the Boston fern right here. And you see this is a gametophyte. This is a little simple plant that is in the life cycle of these ferns, but you see these little filaments. And if you look at these filaments, you see this, they're here. And those are bacteria in there. You see the purple stained, again, for nitrogen. They're getting nitrogen out of those microbes. So they're juicing. These filaments, these hairs that these plants are making, have microbes in them, and they're producing. Okay, so now we move to our more advanced plants. This is one all over the East Coast. You've got it here, the tree of heaven. And if you look, this is a giant weed. It'll grow everywhere, right there. This grows in New York City and little sidewalk crevices or where there's a tiny bit of soil, it'll grow. So this plant is really tough, really capable of growth. And if you look at the leaves, they're covered with these hairs, these filaments. We call them trichomes, right? Plant hairs. But they're just that. Trichome is nothing magic. It's a structure on a plant where they cultivate microbes and juice them for nitrogen. Okay, it's just a filament though. And if we look at it, if we go to the next, this is what these filaments look like close up. You can see all these dots in there, those are bacteria in there, and this is the hair. And you can see bacteria spilling out of pores. These have pores on the side. And the microbes are cultivated underneath each of those, and we think that's where the nitrogen is produced. But the holes there allow some of these bacteria to spill out of these hairs. So they spill out and they'll accumulate on the surface of the leaf. So a lot of the microbes that come on the leaf, that phyloplane, are actually from inside the leaves. They're not, some of them are from outside, but many of them are from inside. They come from inside. They're spilling out of these plants. This shows that actually one of those trichomes, and you can see the motion. You can see these are the bacteria that are traveling, that they circulate them around, like those root hairs, similar type processing. And you can see bacteria coming out. They're coming out of pores here. You can see some of those. And over here, you see some up here too. And even there. And if we had a good image of the side of that, you could actually see holes and see them coming out of the holes. We can watch them coming out. So we know we can make a connection between what's moving in there and then what goes out there. We can actually sometimes see them moving out and then reforming their cell walls. So we can make that connection real easy. Well, this is just one of the chemical reactions that happens. I put this mostly for soil scientists, because they like their chemistry. And so I put a little chemistry in here. But basically what this shows is the plant is producing superoxide. And that superoxide, if the microbe didn't have some defense, that superoxide would destroy it, the microbial cell. And so these microbes secrete reduced forms of nitrogen as antioxidants. And that, for example, nitric oxide then will combine with superoxide, and then we'll get this reaction that forms this intermediary, and you get nitrate. And then, of course, the plant can absorb that nitrate. Okay, so hemp leaves have some other kinds of trichomes. The glandular trichome that you're very familiar with. And like this, looks like this, at least one type looks like this. And you can see it has a stalk on it, and it has a head. And one thing that's notable is this stalk is kind of green. It's got chlorophyll there. This head is not. There's no chlorophyll, no chloroplast there. But it's full of bacteria and chemistry, as you know, and some plant cells too. And the important thing is that we think that these stalks produce the sugars that go into the heads that feed the bacteria and enable them to fix nitrogen from the air, because that's a highly energy dependent process. And so they fix that nitrogen, and then the plant will coax the nitrogen out of those microbes. Yeah.
Is there anything that shows us any differentiation of population or diversity between sessile, captate, non-captate, or is it sort of across the board that they're all having similar microbes?
We don't know. We don't know the diversity of microbes that go into these trichomes. There are differences in, I'll show you a couple of different kinds of trichomes that we have on hemp, but there is a lot of variability in those. You know, some of these glandular trichomes will have huge heads, and so there is this huge variation, which you guys probably already quite familiar with. Okay, what we're looking at here are the very young trichomes. So it would be potentially development, right? A developmental phase where we get this happening. The older trichomes, we can see bacteria there, but they look like they're more filled with oils or something else, you know, cannabinoids or something else that's in there.
So secondary metabolite.
Secondary metabolites, they seem, yeah. Well, the secondary metabolites, it's interesting. We think those secondary metabolites in the cannabis glandular trichomes are oxygen scavenging. A lot of them have antioxidant properties. What we think is happening is that the chemistry, cannabinoids, terpenoids, will sequester oxygen and remove that oxygen from the microbes, make it so the microbes aren't exposed to it. So reduce that oxygen, and that would then speed along nitrogen fixation, because that's what we think. Of course, it's very early in studying that, right? But that's what it looks like. That's the best assessment that we have for the role of those, as opposed to insect defense or something, whatever people think. We think this may be, it's a hypothesis. Yeah.
Have you been able to do any speciation of the bacteria? Do we know what any of them are? I know you said
We do, and I'm not going to talk about it. I have my graduate student has that she has some isolated and are working with. Yeah, some of these are very difficult to work with, you know, because they're microaerophilic. They don't grow with oxygen very well, so you know they're hard to isolate and so forth. But we have some microbes. We know what they are. I'm not going to go into that. No, probably every plant is a little different, right? Because these microbes are all, they can do similar capabilities. There's no magic microbe. You know, they'll get them, you know, if they can. Okay, so this is one of these, this is looking down on a glandular trichome. And you can see in the middle here, you can see we're looking underneath at the stalk, that green, that's the stalk. So we're pressing down on it. You can see the clusters of bacteria here early on. These are in broads. This is superoxide around them. These are they're stripping the cell walls off of them. They're loosening their cell walls. They're forming these trichomes percolate. The bacteria percolate in these. They're not like the ones, filamentous, where they go around and around. Cyclically, these percolate in these. I assume this is the way it normally works, but this is what we see in these trichomes. We see them. And this is a close-up that shows a little more developed. You can see the plant cells in the middle. There's 8 of them. You can see the green underneath. This is all microbes. That's all those protoplasts out there. And you see the interface between the plant and the microbes. You can see this blue, that's superoxide. This is stained for superoxide here. So this is superoxide at this interface. So the plant is, in this interface, juicing these microbes to get the nitrogen out of them. That's what's happening there. That's how we interpret that. That shows that percolation again. That's looking down. And you can see this other kind of trichome here, filamentous trichome. So you got more than one kind right there. Yeah.
Are these videos in real time or are they sped up?
About twice the speed, something like that. About double speed. Yeah. You can see them underneath you can look under the microscope and you can see this motion. Yes. Ben.
Do you have anything that suggests that potentially, are these microbes coming from the environment and then infiltrating into the trichome at the cuticle layer, or is it being introduced through the vascularity of the plant, being brought from the soil up through the matrix of the plant and then
The latter. The latter, from the soil, brought up from the phloem and the xylem, accumulates on these growing shoot tips, on the shoots, the growing shoots, makes a little biofilm on those shoots. Those bacteria are pulled into the cells like they are in the roots, and then they go into the epidermal layer. And in that epidermal layer, they'll do their thing, end up triggering leaf hair production, trichome production, and you get them forming there. And so they're from the soil. The plant is very much pulling these microbes out of the soil. Now you have some on the seed. So if you sterilize that seed, you're going to be sorry because you'll lose some important stuff and it'll take a while for them to get them back. But these are all soil microbes. So they get them, from the soil. Yeah.
Talking about the endosymbiosis and going back to Tim's question earlier, would it be safe to assume or speculate that seeds bred from plants raised in these systems are going to definitely do better off than seeds bred in systems that are missing this biology?
Yeah. It is. These are,
Hydro systems.
So these are, that's a good question. So there is this phenomenon of nutrient density in plants, right? Nutrient density, I mean not only like sugars, but I mean antioxidants that'll be there, nutrient, calcium, other kind of, nutrients, fertilizer type nutrients in plants, right? How rich are they in those nutrients? When they have bacteria, they are rich in nutrients. If we remove the bacteria from them, that plummets significantly. Now, where exactly in that range do we have our hydroponic and aeroponic cultures? They're somewhere in there, right? One may be better than the other a little bit, but it also may be possible to cultivate the plants in such a way that, for example, if you were in an aeroponic system, if you were to put microbes on the shoots, on the leaves, it doesn't matter what's happening in the roots, right? If you could get those microbes, say, with a little compost tea or something, you could spray them on the developing shoots as they're growing early in development. You can get some of those microbes to go into the leaves, and then you'll get some of them into the trichomes, into the glandular trichomes. So you could play with the system and put microbes in it and affect the quality of the crop, depending on where you put it, the quality of the nutrients in the crop. You could change microbes. We've done experiments with carrots, where we took microbes from other plants and then put them on carrots. And depending on the microbe, we increase the amount of carotenes in the plant significantly, 2 or 3 times what we had. Same thing in theory, you could do that with cannabinoids too. You put the right microbes there on the leaves, and you may detect that with certain microbes that you apply, you're going to get a different selection of terpenoids that form. You'd have to do it. I just said it, right? But that doesn't mean it would work, but it should work. It should work. It worked with other plants, yeah.
Do you see, sort of piggybacking off your question, if we're seeing plants that are in bacterially or microbially rich environments, and then we're seeing plants that are in microbially, we'll call it desolate environments, is there, I would assume, the level of epigenetic influence upon the progeny down the line where do we begin to see epigenetic shift in the desolate situations where they are no longer forming these relationships like they're supposed to be because they're so removed from it?
Yeah, what we see, for example, in corn, I can give an example. In corn, what happens is we've been using fertilizers so long that the corn has lost a lot of the microbes, and even the growth of the corn is, the roots in particular are not the same as they used to be. What happens is the roots in chemically liquid fertilized corn, the roots go long down into the soil. They follow the nutrient stream, right, down into the soil. When the microbes, when they're getting nutrients from microbes, the roots change. And what happens is the roots stay in a shallow zone, branch out, much more branching in this shallow zone, aerobic zone, where it can get oxygen, right? That's where those roots are going to grow. But with liquid fertilizer, that's not important. It just goes, those roots just keep going down. So yeah, you see it, you definitely see a difference. Are they permanently losing them? I don't think so. I think we could, what I've known, we can put those microbes back. We could teach those plants to grow properly again. Yeah, I saw your hand.
Yeah, that weakens, also, the root structure weakens the actual strength of.
Yeah. These plants that you feed with liquid fertilizer are weaker. No doubt about it. Plants, they're oxidatively susceptible to stress, right? The oxidative stress, any kind of stress, they're weaker. Plants that you're growing using soil biology, they're going to be tougher plants. They're going to respond better to stress and disease too. It makes a huge difference.
So you had said that in systems where we're utilizing micro-desolate environments, such as traditional hydroponics, stuff like that, you said that you could utilize in early stages of development a foliar application.
You could do that, yeah. That would make sense.
Now, in a soil situation, or even a soilless media where we've got good microbial populations, we're not having to reapply consistently those microbes to the plant for them to have these relationships and begin to do propagation inside the plant. Do you have to do, however, consistent applications of foliar in the microdesolate environment, since there isn't that root matrix construct where they're able to live, survive, and trade and create communities?
I think you do. Yeah, I think you do. I think the mentality of it, you would have to say it's not going to be permanent, but you're going to get if you're going to put something on the plant, the foliar of a compost wash or something like that, you put it on, you push the plant a little bit. Those microbes may degrade, they may not last a few weeks, or you may have to put some more on to get another. If you do something early on, it will have effects that go through the season. That's one thing. So you may not have to keep doing through the season. But the point is, it may not be permanent. People are pushing plants. They're trying to figure out how to push plants. That's one way to push plants without all the fertilizer, is using those foliar treatments. Yeah, and microbial foliar treatments work pretty well too. In fact, you can even put tiny amounts of like algal extracts and micronutrients on leaves and have a big boost. And what we look at the plant tissues, then we can actually see more bacteria in those leaves that were treated like that. So it really does have an effect. The microbiology is there. You can push that microbiology in the leaves just as well as you can push the microbiology in the soil. That's an important principle, and how you could push these plants if you needed to. I already said that. A lot of those terpenoids may be antioxidants. A lot of them are reported as antioxidants. Here's another plant that is in the Cannabis family, and this is hops. Again, my student doing that. This is the glandular trichome of hops, and this is stained for nitrogen. So you can see this trichome is just filled, this purple is nitrogen. And you can see these cells around it on the scale leaf. They don't have all that purple. They become loaded with nitrogen. Curious parallel is that there are parallels between glandular trichomes and root nodules. Interestingly, one of them is that we think nitrogen is being produced we know nitrogen is produced in both, based on what we see and based on what we know already. The other thing is the way they start. They both start from essentially modified plant hairs. A glandular trichome, starts from a trichome like this, and this thing has some bacteria there, but this thing will loop around and will form this kind of a structure, become multicellular, And then when it's fully formed, then it will have bacteria there, and you'll have nitrogen there. Whereas a root nodule will have a root hair, another plant hair, that then will branch, loop around, like I said, and then differentiate into an actual nodule, a bigger nodule. Okay, both start from hairs. That's the point. So there is parallel. I mean, we think they're really different, right? One's on the root, one's on the leaves, but they're different kinds of hairs. Just because one's a root hair, it's on the root, it's still a plant hair, So to the plant, hair is a hair, I mean, there are those on roots and there are those on leaves, but nevertheless, there may be this parallel developmental pattern. There's these parallels. Okay, so we're working with the plant breeder now, Walter Goldstein in Wisconsin, and he's trying to breed organic corn. And I think this is important just to show this. We saw him trying to breed this. This is Walter Goldstein here, but the Mondamin Institute. He's been trying to breed organic corn for a long time. Some of you may know him. I don't know. He's pretty well known, but we're trying to help him. And it's my graduate student working on this. But if you look at, for example, we looked at land races. She's working on land races. These nitrogen-fixing land races. One of them's from Peru, and this is the one I'm going to show you. This Peruvian land race. Okay, these are grown by the native peoples in Peru, and they put no nitrogen on them. They don't need any nitrogen. They grow fine without nitrogen. That's the point. And we look at this plant. If you look at, for example, here, this is the cob leaf, the shuck, young developing shucks. You look at you can see these cells, the epidermis of the cob leaf. You can see these big wads in here. This is inside those cells. This is inside the epidermis of that corn cob leaf. These are big wads of bacteria in there. And any purple around here is nitrogen. This is field corn, sweet corn, modern sweet corn. And you can see, here's the epidermis. You can see the convoluted walls there. And you can see there's definitely bacteria there. We have bacteria there. But they're not as good as those bacteria here in that they put a lot of nitrogen on these plants. There's not as much bacteria in there. There's not as much nitrate that we see coming around. And if we look at the surface of that cob leaf, you see this. These are trichomes. Those are the hairs on those cob leaves of that Peruvian nitrogen-fixing landrace corn. As it turns out, these landrace corn, these nitrogen-fixing corn, have these really long trichomes on them. In fact, some of those trichomes are a centimeter long in those nitrogen-fixing corn compared to the non-nitrogen-fixing corn. So some people are going around looking at their corn to see if they can find the ones that have the longest hairs, and those are the ones they're trying to select for higher nitrogen efficient corn. Okay, but here's a close-up of the hair, and you can see these big wads of bacteria comparable to what you see over here, big wads in there, and you see these little ones too. But that hair, those hairs are loaded with bacteria. They're full of bacteria. We can actually move those bacteria into other plants. And I'll just show you that real fast. All we have to do is, and this is, you already mentioned, I forget who it was. Wendy, you might have been the one to mention it, or James. Rediscovering, where we're rediscovering what native peoples were doing, right? Well, this is kind of what we're doing here. The native Indians in North America had something called corn medicine. And what they would do is when they planted their corn, they would go out into the woodlands, and they would get wild grasses. They would bring those back. They would take the roots, and they would pound them up in water. They would heat that a little bit, which would kill some of the bacteria, but anything with endospores would survive in there, and bacteria that were protected would survive in there. And then they would let that cool down and take their corn kernels, and they would germinate it in that water till the root came out just a little bit. got colonized by bacteria, but they didn't know that. It was magic. Okay, they would take that slightly germinated corn, they would take it, plant it in their mounds or plant it in their fields, whatever they were doing. And they did that routinely. You know, it's published exactly how they did it. And it was magic to them. It was magic. But of course, it's microbiology to us. We did that with that corn, ground up that corn in some water, took the water, put it on another plant. In this case, this grass. This is a bluegrass. This is where we put that landrace water, okay, with the bacteria in it. On this is bluegrass, and here's where we did not. This is just water treated. You can see these are a little bit bigger, not much though. But that wouldn't convince you at all if that's what you. Okay, maybe this. Okay, here's the root where we did not put it. You see there's some hairs there, but not that many. Here's where we did put it. You see now there's a lot of hairs. Okay. Now this is the root cap cells that sloughed off. Okay, so here's where we did not put it. You see there's bacteria there. These little bitty dots all in here, definitely bacteria. Okay, here's where we did put it. Now you see the big bacteria that were similar to what we saw in that landrace corn. We got it to go in, and this is stained. Both of these were stained for nitrogen. So the blue is nitrogen. So all of that is nitrogen that was being produced in that landrace corn. So we can take those microbes and move them. We could do this to any crop. Do the same thing. Someday, maybe 30 years down the road, there's going to be selections of corn or any kind of crop that's going to be enriched with nitrogen-fixing microbes to reduce at least that's the idea, right? That's the hope that something like that can happen. Here's the root hair, and here you can see this is the bluegrass where we did not put it, and you can see there's some bacteria there. You see some little rods in there. Those are bacteria, but it's not blue. This is stained for nitrogen. Both of these were stained equally. Now here's where we did put it. Now you can see the root hair, and you see the blue there. That's the nitrate that can, and you can see where it is too. It's at the tip where we said it was, at those root tips, that's where the nitrogen came out. And that's where it came out. You can see them running it down the side there, but that's where it formed. And this is 30 days, 35 days. This is the one that we transformed with that landrace corn bacterium. And here's where we did not. And you can see they look different. That held up for a long time. Now these plants are sitting on our back patio. My wife can vouch for that. They're there. They're all looking pretty dead, but hopefully in the spring we may see some recovery on hopefully the one where we put that. Hopefully that one, but we'll see. Maybe it's just frost, just dead. I don't know. We'll see. One of the important lessons here is that these microbes are all over the plant. They're in the roots, right? They're getting nutrients in the roots. They're in the leaves, on the leaves. I said the trichomes on the leaves have microbes in them. They're on those cob leaves. We think that the cob leaves, the microbes in those cob leaves actually produce the nitrogen that fills out those kernels, that enables those kernels to have protein. And that's what we're finding in this breeding program, is that these lines have richer, nutrient-dense, protein-rich, more methionine, more antioxidant amino acids than in these conventional forms of corn. So, okay. Wendy, how am I doing? Do I got a couple minutes, or should we do the thing? What do you want to do? So keep doing? Okay, well then I will go to the fungus part, because this is of interest to you, I think.
I actually have a quick question that's
yes, Wendy.
bringing something in. So when there was a young lady talking about, testing for cannabis in Massachusetts and in a lot of states, and they're doing these total plate counts.
Right.
So I'm kind of like, is there almost like a scientific basis to say that you can't do total plate count because we need it in order to produce the plant in general?
That's what I was chasing after, and I was
Yeah,
asking him about
I'm like-
specific bacterial strains, and I was like, if we can prove that some of the bacterial strains that they're testing for with CFU units are naturally occurring within plants, then completely defeats their entire.
Well, in time, we might be able to prove that.
In time.
In time, we might be able to prove that, yeah.
I'm just saying as a generalization right now, we don't have to get into the deep science of it.
Yeah. Well, they're all in here, so it's kind of ridiculous to declare these plants are no good because there's microbes there, because that's how they grow.
Right.
You know, this is normal. This is how they work. Now, I know I've visited A&L Canada Labs. They do a lot of screening, and I didn't know they were doing plate counts, but I know they were screening for specific strains of microbes that were pathogenic, right? My understanding is that was the main thing they were doing in Canada, but I didn't get into that deeply enough.
Yeah, Canada, I believe, is that, and so is California, and that it's just like certain aspergillus and E. coli.
That's
and,
better.
A few just general ones. They're like, these are dangerous. As long as those aren't present,
Yeah.
we're fine. But a lot of the states are taking total yeast and mold counts, total aerobic plate counts. Colorado is another one. They're total yeast and mold. Like,
They should look at broccoli. They should look at broccoli. Broccoli and cabbage, the brassicas, are so full of microbes. When we put them under a microscope, they're swimming literally inside the cells. You know, cannabis, we don't see that. I mean, yeah, there's microbes there, you can see it. But if they go to the cruciferous crops, there's a lot more bacteria in there. And I'm sure if you did a plate count on the brassicas, you'd probably throw them. It's through the roof. You'd throw a lot of them away if that was the critical determinant there. Yeah. Okay, so the other thing that I want to say, because I am a plant pathologist, you know, fungi will also internalize bacteria. And when they internalize bacteria, their behavior changes. In most cases, for example, with the pathogen like Fusarium, which can cause a lot of problems, when the bacteria are present, it doesn't hurt the fungus, the bacteria don't, but it changes their behavior. They lose their pathogenicity. They become avirulent. You know, understanding that is, we don't fully understand it yet, but it's a phenomenon that's really interesting, because it means that we can control fungal diseases using bacteria if we know how to do it, which bacteria, how to apply it, and so forth. And there's well, anyways, this kind of shows some of that. There's a bacterium in the middle here. And you can see this out here. Those are all the fungi. And there's hyphae, there's mycelium in there, but they're covered with bacteria. And they're not sporulating. I mean, you can see the other one doesn't. You got the bacteria there. It has no effect on the fungus. But this one changes the fungus. And this shows it again. Here you can see, this is soil. And you can see some seedlings here. This white cobwebby, this kind of white color, that's all fungi growing on that. There's no bacteria here. We removed the bacteria. These are actually being colonized by that fungus too. That's a Fusarium. Here is the Fusarium plus a bacterium. This is actually a Pseudomonad, very common soil plant bacterium, very common. Put it in around the plants, and you see there's no fungi growing on the surface of the soil. These seedlings won't have any disease. They're unaffected by that fungus. Totally unaffected. We can do experiments with something, for example, basil. Basil, a little seed that has a little gelatinous coating on it, and that coating is full of bacteria. If we take Fusarium and put it with the basil, it does nothing to it. It won't affect it. If we take some Clorox or Sterilant and we remove those bacteria, put Fusarium there, it eats the seedlings up. they have to have the bacteria are part of the immune system of the plant. And so if we impact that, these plants are sitting ducks. Okay, that's why many of our agriculture, we have such disease problems in agriculture, because we've lost a lot of these microbes. That's one reason. There's lots of other reasons that people give, like monocultures and so forth. That's all probably related to there. Okay, but the biggest issue is because those microbes have been lost. And these plants now are... Now what happens to these bacteria? This is a hypha of a fungus. And these little dots, those are the bacteria. They're actually taken into the hyphae of the fungus. They go in the hyphae of the fungus. Now, are these fungi getting nutrients out of these bacteria? There's some evidence to think that they are. We have some with some of the fungi. We can actually see nitrogen coming out of the bacteria into the plant. So with that idea, we think that, well, if these fungi are able to get the nutrients they need from bacteria, maybe they don't attack the plant. So it's a kind of a principle that's uncertain, but it's kind of interesting to play with that idea. Maybe it's a new way to prevent disease by satisfying these fungi with putting microbes there. And here, this actually shows a hypha. You can see the bacteria actually coming out. Here too, this is what it looks like. Here you can see the hyphae there in the middle, and you see the bacteria coming out. When they come out laterally, they'll make a little hole, and then they'll make their rods like that. So they always look perpendicular like that on the hyphae, so we can pick it out. Same thing on a root here. They'll do the same thing. And so you have that perpendicular kind of pattern to these microbes. And this actually shows it. This you can see this arrow. You can actually see something's coming out through the hypha wall, and you see it kind of branches here, and you have a cell here and a cell here. That's where the bacteria came out of the hyphae. And so we can track it and see what it was in, what it was out. Sometimes we can see at the growing hyphal tips, we see them going in too. Ruben.
Is this true for all fungal hyphae, or is there a difference between...
It's a good question, Ruben. There are different groups of fungi. And we know, for example, that some fungi are obligate biotrophs, right? They will live mainly like rust, for example, right? Rust fungi or powdery mildews. They will live only on their plant. They don't grow in the soil. They live only on their plant or on a susceptible host, and they put little hostori into the plant to suck nutrients. Those are really specialists, specialist, parasitic specialist fungi. But many of other fungi are partially soil microbes. They are facultative pathogens, right? They also live in the soil. They'll degrade plant material. They'll eat bacteria, possibly, and they'll cause disease. So what happens is with those fungi, we can favor the soil non-pathogenic phase by having bacteria there. We don't know about the rusts and the smuts. We're curious about those biotrophic, obligate biotrophs, about those. I do know that there are ways that people can control them. And I don't remember if they're using microbes or not. Vaguely, I think so for some of them, but that's an area of uncertainty, Ruben. It's a really good question. No, it's not the same for everything, but yet there may be places on some of these other groups of fungi where they do get some nutrients from microbes, and so we're interested in that, curious about that. Yeah.
Could you tell me again what the specific strain of soil bacteria was that you were utilizing in that fusarium experiment? I know you mentioned, I just didn't catch it.
Pseudomonas. That particular one is a Pseudomonas fluorescens, I think. Yeah. It's that LB4, I think it's Sandy LB4 actually it was one that came off of, who was that? Yeah, there it was. It actually came off that reedgrass plant. So we took it off reedgrass and we put it on in this system to work with it. They're common. A lot of these microbes just go from plant to plant to plant. They're not adapted to any one, but they adapt to multiple. There's only a few that are only adapted to a specific range of plants. Many of them can go in multiple plants. So plants can actually share microbes, you know. Okay, so you saw that. And this is the final slide, just to summarize things. So rhizophagy gives plants nutrients, lots of soil nutrients, right? And they're getting nutrients through nitrogen fixation in the plant, too, in different parts of the plant. The other thing is, because it's an oxidative interaction with these microbes, plants become resistant to oxidative stress. So they're hardier plants. And finally, the soil pathogens. They are part of the immune system of the plant, and they protect the plant from all kinds of diseases. We normally think that when someone has a disease problem, a fungal disease problem, they usually resort to fungicides, but actually the better way would be to put more bacteria on those plants to protect them from the fungi. So this guy wrote a book. By the way, this guy right here, Jeff Lowenfels, he wrote this book, and it's teeming with bacteria. He has several others. He has teeming with nutrients, teeming with microbes, teeming with fungi. There it is. There's the book right there. There it is. All right. All right. And this is Teeming with Bacteria, the Organic Gardener's Guide to Endophytic Bacteria and the Rhizophagy Cycle. So in writing this book, he told me already this is a bestseller for garden books. So it's already very popular. And so more and more people are learning about endophytes and the Rhizophagy Cycle. And it's all Jeff that did that because he did this book. Yeah.
His books are kind of our Bibles. You know, the cannabis community, those are our
Yeah.
Bibles pretty much, yeah. Teeming with microbes. Yeah, now it's 4 books.
It was a trilogy, yeah.
For a long time, yeah.
Now what is it? What is it?
Now we're working on the penta.
Quadrology, or what is it?
Quadrology.
What is this? Trilogy, what is it for? Yeah, so, and this is a lot of people did this work, and a lot of people get credit. I didn't get to mention everybody, but this is almost everybody mentioned here. I think I might have left one of my graduate students off. And the last talk I did, I think I made him I might have upset him a little bit, but he does have a picture. His picture now I showed on one of the early ones on a tomato. I just don't have it here. But so that's everything that I have to do. So any other questions? I see right in the back, middle.
So dead soil that has no biology, we're trying to reintroduce inoculants that contain what we believe to be beneficial bacteria and fungi. Is that doing any good?
I won't say no, because it may, especially if you put a plant right there, right? And you put it on that plant, and so you're going to get a response. But I think the best solution, and you know it already, is to build that soil up, to build the organic matter, to try to have cover crops, living cover crops on a soil. And a diversity of cover crops means, because every plant has different microbes in it, so a diversity of cover crops means you're going to get a diversity of microbes going into the soil. And as you build up that organic material, you're going to build up that microbial population, and that's the best answer. Yeah, you could help, but I think in the long term, there's no way out of rebuilding our soils. There's no way out of that. Yeah. Here, and then I'll come back.
I was reading this book, Hemisphere, and he talks about making chlorophyll water, where he essentially takes plant leaves and blends them. And I always thought of it as a great way to, I do it in my garden, and a great way to bioavailable nutrients and enzymes and all sorts of other things. But from your talks, it could also be a biological inoculant from looking at what the Native Americans used to do with and stuff. So
they were doing it. Yeah.
Yeah. So using diverse different types of leaves and species could be bringing in a bunch of different types of endophytes and inoculating the plant.
I really believe that this is a way that that growers can experiment and possibly come up with some new methods improve the crops by putting these microbes from other plants on them. I really do think that.
I know there's great results by doing it. It's
Yeah.
amazing what it does to plants.
You do it, and it,
I do it.
yeah,
Just take comfort leaves,
and it works. Yeah, you're getting microbes there and nutrients and everything. Yeah,
stimulating everything that's
yeah,
there.
just like the Iroquois, the Native Americans. Yeah. Now I saw him, but let me go here first.
So you
yeah.
said earlier that kind of like the super endophytes would transfer to the top,
Yeah.
where some would go up,
Mhm.
the majority of them would go up, like the best of the best would go to the top.
Yes.
Are you referring to like go to the seed?
Ovule. They'll go to the ovule, they'll go to the leaves. They'll go. The reason they're growing on that shoot meristem is because they move up through the, they don't have far to go when the seedling starts to grow, right? They don't have very far to get there, but they go there to that shoot meristem, and then they start growing on that shoot meristem. There's lots of sugars on that shoot meristem, and so they're, in a sense, the plant is likely cultivating them there intentionally, right? Intentionally, and then taking them in. We can see them going in on those leaves, on those developing leaves. So this is actually how it naturally works, how that system naturally will work.
So what my thought process went to, can we foster that?
Yeah.
Cultivate, grab those things that the plants already cultivated, back to the endophytes, and then reintroduce them into plants that might need a super endophyte.
You could. You could. What I would do to do it right? I mean, I would go and you want to get the youngest shoots just before they're very green, right? When they just come out, you know they're really soft and tender. Get those, get the youngest ones, and then grind those up, put those in water and use that. Or if you are so inclined, you could isolate them out and then put them. The problem is, a lot of these microbes you can't grow, right? You won't cultivate. They're not cultivating easily. So you can't, it's not like you can reliably isolate it and then use it. Just some of them you're not going to be able to grow out, either because they're microaerophilic or something like that. They can't take the oxygen. So you'd have to use your method. You know, if you had a reliable seed supply and you have it growing well, and you have some other varieties that aren't so good, you could take that best one, pull those shoots off of that, grind those up, put them on seeds, say grow them in a rag doll or something, germinate them in a rag doll, right? Little rolled in paper, put your triturate of your plant tissue all over it, let them germinate, then take those and transfer them. Compare them, of course, to a control. You always have to do a kind of a simple little experiment, right? But yeah, it would work. What you say would work, and you could improve it should work. You may have to play with concentration, you know, but yeah, it should work. It would work good. Okay, here?
So I'm not sure if I remember this, but in the nutrient extraction zone of the root shoot there, you mentioned how like the superoxide sometimes either fully, partially, or not at all degrades that bacteria in there, like the cell wall of it. I'm just wondering, like, what happens to the non-degraded bacteria in there?
They become a problem if they take in. Now, usually they won't take in bacteria that are too resistant. Usually they don't take them in for some reason. I don't know why they don't take them in, but usually they don't take them in when they have a lot of other microbes there, right? They're competing with. If we remove all those other microbes and we give them only one that is oxidatively resistant, they'll take it in. And then when it goes in, what happens is that microbe will replicate out of control. And the plant growth is suppressed. And so we'll see like shorter roots. We'll see bacteria that come out all over around the root. And the seedlings just don't develop properly. But that's not a normal thing. That's when we force a microbe to go in that's not adapted to the plant. I mean, that's maybe significant. For example, we're playing around with trying to create some organic, edible bioherbicides, right? So we're taking some of those resistant ones and we're trying to formulate them in such a way that we could control weeds with using that kind of a microbe. Yeah, but I don't know if that's going to work ultimately in the end, but that's what we're trying to do. But it's hard. The plants won't normally take those microbes in that are more resistant. Usually, how it selects, I do not know.
So just to cap on that, I had a question with
Yeah.
You're saying that you're looking to utilize essentially a reverse engineered biostimulant. You're intentionally weakening and sickening the weed plant.
Exactly. It is like that. It's a biostimulant that weakens instead of. Yeah, it's crazy. We actually have a fancy term. Academics, right? We have to make up words that endobiome interference, right? Doesn't that sound cool? Endobiome interference. Well, yeah, that's the term, but will it actually work? We don't know.
So my question that I wanted to pose to you, which you alluded to a little bit,
Yeah.
is if we're looking at this soil bacterium and we're looking at the way that it interacts within the plants, within the fungi, the way that we're getting exudates and endophytes reacting, but we know that we can only populate in labs specifically very, very small portions of these microbes. What is the best methodologies that you could recommend for application at scale? Obviously, in a small application, you can gather small amounts of plant matter either from your already healthy cultivation or from an exterior source, blend it and apply it. But if we're talking a scale, we're talking a vegetable garden that's a couple hundred acres, we're talking a cannabis cultivation facility.
Right.
How can we apply this in a manner where we're not just doing, the selective two bacterium that we can make in a lab, but we're also not just going out with the paintbrush effect and grabbing soil and just going for it.
Or grabbing a lot of plants and grinding those up and putting them on. Although that may not be I know it would cost a lot in cannabis if you get all those all those shoots, right, that are flowers, inflorescences, and ground them up, you probably lose a lot of money. And it would probably work, but ultimately I think, there's, to scale, it may be difficult unless we could find out exactly what the microbes are, get them to grow out, and then take those a lot of these are going to be gram negatives, and so you may have to microencapsulate them or do something to get them to where you can apply them, right? Grow them out and apply them, spray them or something like that. That's that's going to happen. The companies are already trying to do this. We talked about bug in a jug. They're trying to do this or that. They're doing things like genetically manipulating microbes to try to get them to fix nitrogen. I don't think it's working very well. The problem is they don't know. We don't know how. It's just a shotgun. Yeah, it's a guess. It's a molecular biologist's guess. And business guys' guesses about this is going to work. Okay, this is going to work because we're going to turn off the repressor genes for nitrogen fixation in our microbe. Then we're going to give it to everybody to put it on their plants. The problem is that I've heard their studies aren't going so well.
Yeah, my next question would be, what would you think about diversity of application as far as die-off rates? I mean, microbes are so tailored to the environments that they live within. That's something that you cultivate.
It is really, if it's microaerophilic, you're not going to be able to do that. Pan spray, there's oxygen everywhere, it's not going to go. We have to do our experiments, our isolations in low oxygen environment, in high nitrogen environment, right, without oxygen. So isolate it without oxygen, and you probably have to infect the plant without oxygen. So that makes it even trickier. So to scale, it might be hard in some of those cases. Yeah, it may. I saw a hand over here.
Could you do it through process?
You can, but you can't do it through scale, right? We can do it through a single seedling or through a bunch of seedlings that we inoculate. But then you've got to, it's like, the system, I think, you have to breed in to the plants. So you're going to have to work with a plant breeder, right? Put them in, try to stabilize them, make sure they're in there, figure out how you have to treat them to grow them, right? How you have to treat them so you don't lose them like that. I think it's going to be more of a gradual kind of effortful process to get the microbes back into our crops. So it's going to take a long time. Yeah, Leslie.
Okay, so I'm going to ask us to zoom way out for a moment. Massachusetts recently released a report called the Massachusetts Healthy Soil Action Plan. In fact, NOFA was one of the authors of it.
And the point of this report was to take a look at all of the different types of landscapes that are in the state, and based on using soil organic carbon levels as kind of an operating principle by which to estimate where is the soil the healthiest, whether it's in agriculture, forests, wetlands, lawn and recreational landscapes, or even impervious landscapes. And therefore, by knowing that, plan for where nature-based solutions to address climate change and biodiversity loss could be most effective. And so, what do you think about the whole notion of using soil organic carbon levels alone as a kind of a screening tool to define soil health?
Well, it's very important, right? It's very important, but if there's, if you have good soil organic carbon, you likely have a history of good microbial activity and plant growth in that area, so it correlates. I was going to say that if there's some toxins or something in that environment, you may not pick that up if you're only looking at organic carbon, right? If you're only looking at organic carbon levels. But it may be, that may be acceptable now. It may correlate quite well. Yeah, I can't say. I won't.
Well, I guess it was a case where the data were available, and we know that wetlands have the highest levels of organic carbon because it accumulates in
Yeah. That's not a.
forests or next.
Yeah. That's not. Yeah. Well, wetlands, you don't have a lot of oxygen in the soil, so it's not going to be a particularly good environment for plants.
Right, but it's a great place to
Sequester stuff, yeah.
So in terms of drawing down
Would not.
from the atmosphere, it's great. So it undoubtedly it has some utility. I was just curious about, is it one tool, I guess?
It's one
I
tool.
don't know anything about that particular soil organic carbon, whether it's really a healthy biodiverse soil or just a place where a lot of carbon accumulated.
I'd have to study it more, wouldn't you? Have to look at it more.
Oh,
Yeah, have to look at it to conclude.
A lot of us are looking at it.
Yeah.
Thank you.
You're welcome. James, you had your hand up.
Yeah, well, I was just wondering, I heard you mention a few times diversity, and we were talking a lot about identifying, isolating microbes, and obviously talking about the fact that a lot of them don't grow in the lab. Are we moving to an area where we're eventually going to get to a point where the answer is this is as much as we're going to ever know, and that we can do as much as we possibly can to bring diversity in using the tools we have, but maybe is there a wall that we hit? Is it possible that we're never able to grow those microbes in the lab, so the only way to get them there is through just natural means?
I know we've not hit a wall. I know we have not hit a wall. As we see what's going on, we're learning little bits how to manipulate the system and how the system works. So I think that in time we're going to be able to grow a lot of those things that we can't grow. We'll figure out methods, like I said, the high nitrogen environment. That's a trick for isolation, also a trick for infection, right? No one uses that. But, we can see the microbe, we know it's microaerophilic, so we know how to work with it, right? We could play with it. As people see these systems and we're able to manipulate them, we'll learn more about them. So I don't think. Now we may have hit a wall with our attitudes about agriculture, right? We may hit that wall and realize now, wow, plants don't work that way. That's kind of a realization that if plants are doing this is how they get their nutrients, this is how they work. Why aren't we encouraging this? Why aren't we doing this more? This is not just this fundamentally changes how we think plants work. It's a fundamental change. They are microbe internalizers. They're microbe farmers. That's fundamentally different than what we thought before. What we thought before is the microbes are out there, they're just kind of hanging around, doing stuff in the soil, but doesn't have a lot to do with the plant. They'll get some more nutrients and the plant can absorb that, but the plant's not farming them in that sense, certainly not absorbing them into their tissues and processing them and getting nutrients out of them. So this is it's very different. And, our old ideas about how plants work, that's hitting a wall. They don't work like that.
One more question.
Yeah.
So,
I see it.
as a natural farmer, I feel like I would be remiss not to ask a highly educated plant pathologist what his opinion is after talking about all of these different microbes, how they work together, how we're able to utilize them, the fact that we're not able to propagate a vast amount of them, and that we don't really have an application at scale of how to gather these microbes and propagate them out and apply them. Are you familiar with natural farming, and are you familiar with indigenous microorganisms and how we capture them and propagate them? And if so, what's your opinion?
Yeah, I'm somewhat familiar with it. Not the details, but of some of the practices. I think they are practical. I think there oftentimes is a spiritual element to it. And I don't think that's bad. I think that people have lost the connection with nature, right? And that this is an area that we can reclaim that connection. And I can tell, listening to people here talk, you have that connection already. The natural. You have a common belief that this is the right way to go. This is the healthy way to go. There's, I think it's a fundamental grounding, and the spirituality is part of that. So I think it's good. I don't have any problem with it at all. I think we need that to make that connection and to change agriculture. We need to have that relationship with the earth and with plants growing. Yeah.
How would a endomycorrhizal filament look compared to a rhizophagic root hair?
They look very different. Now, I will tell you that the endomycorrhizae are another group where we know that we've had bacteria internalized in them for a long time. And there's some work that has gone out. A lot of the VA mycorrhizae, for example, are just loaded with bacteria in there. You can see the papers about that. They would be longer. There would be melanin in the walls of the VA mycorrhizae. You would have thick and thin areas as the mycelium gets thin in parts and thick. You would also have connection with the roots where you would have arbustles or some vesicles or something in the roots.
You need a microscope to see?
You would need you might be able to see discoloration in the roots without the microscope, but yeah, you would need a microscope to really see the hyphae there.
Because when you're looking at a nice white fuzzy root system, it looks great. You can tell it's inoculated with microbes, but could you tell if it was rhizophagic bacteria or?
Yeah, I could tell.
Okay.
Yeah, you could tell too if you started to study it. Okay. That's why the nice thing about the microscopy is that you could take pictures of things, right? Yeah. Especially the new cameras have now where they connect to a laptop and a little digital camera that you could take a picture of that, and then you could study that, right? Okay. A few images that you could study, and then you could look at the features. I mean, for example, hyphae branch, right? Hyphae are going to branch. They're going to be different sizes. So you would look at those features and say, well, which is this? Yeah.
Would it be common to have both present?
Yes. Yes. It would be common to have both happening. Absolutely. Yeah. You're welcome.
Could
Yes.
that be determined with or without staining?
Without staining. No staining.
Even with our muscular mycorrhiza?
Yeah. No staining. We could see it without it. Yeah. You could stain it and see them better, but we could see it without staining because the hyphae themselves are pigmented. They're brown on those roots. They're going to be brown coming out of the roots. The VA, the arbustles themselves are going to be fine inside the leaves. You're still inside the root cells. You're still going to be able to see them. You just have to kind of know what you're looking for. You can still see them.
So you might need to be trained to actually differentiate.
You may need to be trained. I mean, to see the stuff that I showed you, right? How much training did I have? 45 years or 47 years looking through a microscope, right? Yeah, like that. Yeah.
Are symbiotic fungi taking a backseat to the rhizophagy bacteria, or are they just another ally?
It's another ally. Okay. It's another ally. Yeah. Tim?
So more than nuts and bolts about KNF, specifically IMO. Are you familiar with what IMO collection?
Remind me again what IMO is. I know what KNF is. Indigenous microorganisms. Yeah. Yeah. So they're the best kind.
Specifically, the microbiology that's found in more diverse, less touched areas. So we're talking about compost tiles and backyards, but like untouched forest or field.
Well, that's what I think.
So specifically, IMO is a par-cooked rice that's then placed in a forest system and then is used as a collection for microorganisms.
It's a good idea.
What type of microorganisms do you think a collection like that would capture? And do you think endophytic microbes that could potentially be in the par-cooked rice would play a factor in the potential development of the micropopulation blooms?
Yeah, I think that it's a good idea to capture those forest microbes on any kind of a trap crop. We would call it a trap crop,
But
right?
it's not even, it's actual rice. It's actual
Rice, yeah, I understand.
That are par-cooked and then placed inside of a box. And
Oh, you put them there.
Not an actual living plant.
Not a
just
plant.
Rice
Okay,
grains
I heard
that
about
are
that. Yeah,
what
I heard
type
about
of
that.
collection do you
Yeah.
think that would be? I understand that a rice plant would apply quite a lot.
Well, I'd have to study that a bit I'd have to look at that to be sure. I can't say. I would think that if they're taking soil and sprinkling soil on it, or taking grass clippings and or plant clippings and sprinkling that on it, that you're getting microbes right out of that material, if that's how they did that, right? So yeah, I think that's a good idea.
Actually, since we have some time before Davey starts, we actually have IMO, we have soil, and Wendy has a bunch of pictures of former IMOs. I think it'd probably be great to actually look at them and talk about what's in there. I know she's had some conversations with the mycologist about some of them. I think our opinion is, saprobes, even if it's just saprobes, are good. Yeah.
They carry bacteria in them. So if you have fungi there that are saprophytic, there's got to be some bacteria there that can go into the plant.
So from my understanding and from some DNA research I've seen on some IMO samples, they're all different, they're all diverse. It seems like a lot of mucor and those type of microbes, which from my understanding, are common in food bowls, but they also have great staphylococcus properties and, nutrient properties.
That group, mucor, mucorales, all of those things, they're one of the main internalizers of bacteria. They are full of bacteria. If you look at hyphae, I don't have mine. I can't show you now, but those hyphae of the mucoraceous fungi are jam-packed full, more than in the pictures that I showed you. Those were ascomycetes. The mucoraceous things are really full of bacteria. They're very, very full.
Because they're one of the main decomposers
Yeah.
in the environment, so they'd be very diverse.
I believe those DNA samples are also IMO1, right?
I'm not sure, like I said,
Well, which is a whole different, for those of you who don't know what we're talking about, pre-natural farming, there's one of the staples of CANF is taking biology from diverse areas out in the forests and fields that are untouched by human hands, and you're trying to capture the delicate diversity that's there, especially the different microbiology that doesn't do so well once we start tilling and feeding and doing all kinds of things that we do as humans. So if you can capture those, the idea is you do collections throughout the year, spring, summer, fall, winter if it doesn't get too cold, which most of us can't do. And then you combine all of those collections, your IMO1 collections, into a pile that has carbon and carbohydrate, as well as some other years for microbiology. And you have IMO3 at that point. IMO2 is when you create a shelf-stable version of it. So you have all the diversity from all the seasons, from all the spaces, and you're combining it into this larger pile. Then you can take that to IMO 4, which includes your soil from wherever you're going to grow. And then you can take it all the way to IMO 5, which includes a nitrogen-rich source, which becomes kind of a fertilizer as well. So the debate, and Tim, we talk about this a lot.
Okay.
Tim is in our back group channel for organic cultivators, and we always are battling, trying to figure things out. And he does these
I just want to get to the
deep
root of
dives,
it. I love it.
amazing dives on these like research studies. We'll be like, look what I learned, and we're like, yeah, but that's in a lab setting. That's not out in nature, and I don't know that translates quite so well. And so what he's talking about is there is somebody out there who's doing IMO, and they are doing some DNA research, and it's become very apparent that it's fairly limited. What's come back is it's not the diversity we were expecting. However, looking into this person's research, it was done on one IMO 1 sample, which, okay, you have one little box of rice under one little tree in one little area during one time of the season. I don't expect it to be as diverse as when you take it through to IMO 3, which includes many, many, many collections of IMO 1. So it's kind of this, we're having this whole thing. Can you get endophytic mycorrhizae, in this collection because it's dead rice? And I'm like, for me, I just take a track of like, I don't really know. All I know is that it works really well, and we see some amazing things in the garden when we are doing this stuff, and the amount of fungal fruiting bodies that appear when you start doing green natural farming that weren't there before shows that there is definitely diversity. How much and of what, who knows?
The idea that the mycorrhizae are responsible for the growth boost, it may not be a valid idea, really, that it's the other bacteria maybe that are coming out of these other plants and fungi that are more likely, more capable of boosting the plant growth. So mycorrhizae, when we even look at many of the mycorrhizal products, mostly what we get out are bacteria rather than the mycorrhizae. So I know that people oftentimes say, oh, the mycorrhizae do this. Mycorrhizae are not as important as these other soil microbes for plants. They help, they definitely help. And some plants, trees, for example, have to have the mycorrhizae to get the nutrients out of the soil, right? All over. But for most of our other crops, mycorrhizae are not that significant. They're there, but they're not that beneficial.
Is mycorrhizae more, is the function of mycorrhizae more to elicit a community between the plants? Because we know they can share nutrients and messages, essentially. Is that mainly what its function is?
That's a very modern idea, but the VA mycorrhizae don't really do that. And the VA mycorrhizae that you see, they're, especially in agricultural soils, they're broken apart because of tillage. They're limited in scope. You pull a plant, you harvest a plant, it's not there permanently, and so the mycorrhizae aren't permanently connected. That phenomenon in soil with VA mycorrhizae is probably not, I'm not going to say it doesn't exist, because there's some of it that happens, but most of what we see in soils, in our tilled soils, even in our no-tilled soils, where we put crops in, are bacterial effects, bacterial and yeast, and that's not a small thing. The fungi, for example, the mucorrhizas thing and the fusarium, if you got lots of bacteria there, that benefits plants quite a lot. In fact, with the bacteria there on fusarium will grow as an endophyte in the plant, and the plants will grow well. They'll grow fine. Some of the nutrients are coming from those fungi that the bacteria are interacting with. So the VA mycorrhizae, they're definitely there. They can help, but I think that's not the main benefit of a healthy, diverse soil. It's all the other soil microbes that are there. We latch on to mycorrhizae as more important than I think they really are. Yeah.
Yeah, good question. My understanding, at least of like a community organization, I used to work in a soil lab, like a micro lab for a while, is that essentially it's like largely ectomycorrhiza that are actually forming like large networks. And
Yeah, right. In woods, yeah.
Yeah, and it's like it's not just it's specifically conifer, if I remember
No, there's
Well, those are like
big trees, though. Mostly big trees
Yeah,
do it.
definitely.
Yeah.
And then arbuscular mycorrhizae might connect like one or few plants together, maybe, but it's usually like a
Less.
singular
Less. Yeah.
Also, if anyone wants to look at it, I have some really cool stained and non-stained arbuscular images on my phone from back in the day, but it's exactly what you're talking about. You can still see them without staining them, they're kind of brown. If anyone wants to look at it.
And that's not to belittle VA mycorrhizae. They're definitely there, and you definitely see them on plants. Plants definitely benefit, but not to the extent that they benefit with all these other soil microbes. We can't even get them to infect plants. Usually when we get a product and try to put them on, very rarely do they even infect the plants. Yeah.
In your opinion, do you think that trichoderma is a better product to use?
Trichoderma is a good product, partly because it has microbes in it, a lot of microbes there. The other thing is when you get it out, and some of them are endophytic, and so microbes can go into the plant. Yeah, some of these other fungi, if you put them in, you will see a boost with the plant. Trichoderma is a good one to do, and on top of that, it will sometimes parasitize other fungi in the soil, and so that'll also have a beneficial effect.
I understand it also fixates iron, so you were talking about how important
Yeah,
it is
right,
in the Rhizophagy cycle.
right, exactly. Yeah, I saw a hand back there too.
Oh yeah, just one other question. So in terms of like whenever you're pulling stuff, whether or not be like an IMO or pulling out of nature and stuff like that, one thing that has always kind of stuck is if you're adding a carbohydrate source, it's sort of like similar to agar in the sense that you're not really talking about like a natural ecosystem now. You're sort of like removing the limiting growth factors.
Right.
Do you think that it would be difficult to pull a representative sample from a natural environment when all of a sudden it's what's latching onto this like really easily accessible food source? Do you think it would be all like this? Still get a diversity of that, or would you just get the first to the prize? Whoever's going to grab their repopulate super quickly would be able to take over.
Well, I think if I were doing that, I would get not one plant, but I'd sample the plants and then take the roots and try to get an average of what's there, grind that up, get some material to the inoculate. Is that what you're saying?
Well,
to inoculate?
So like when people are doing like IMOs, for example.
Oh, okay, yeah.
Doing, like, compost
Yeah.
seeds, the first thing you'll see is that they put a bunch of carbohydrates in there.
Yeah.
Which is like removing a limiting factor on growth.
No, you get sugar fungi is what you get. Sugar microbes. You get the sugar microbes. That's where a lot of the mucuraceous things are actually sugar. They're actually sugar fungi, but they're taking microbes from whatever else you have in there. They're absorbing those.
I like diversity, even though you're sort of like, because it's sort of like line create like line effect. It's like whoever's first to the party, you can just like
the diversity is a thing, but what you're also doing is you're selecting a subset of microbes, right? If you're gonna take the say what was the fungi that was growing there and the other organisms that were growing there, and you're gonna grind that up and get some microbes out of it, put that on some seedlings or something. You're selecting a specific environment with a specific set of microbes, and they may be coming out of this other plant material and the fungal material you have there. Those may be better adapted to the plants you're going to put it on, simply because they're already endophytes, right? Definitely. They're already endophytes, so you're getting endophytes out. So you may not have to have a hugely diverse set of endophytes if you have some that are going to work in the system.
So you think it's probably likely to end up with like a few dominant overtakers?
Yeah. Oh, yeah.
Those then will have endophytes.
Yeah, that's what I think.
Which will then.
Yeah, that's what I suspect. Yeah. Yeah. Tim?
So that was kind of like my theory with the IMO, is I feel like it's collecting a lot of the Microbes species and a lot of like the more carbohydrates and simple sugar collectors. But it's probably also collecting a lot of bacteria and a lot of other things. But then I feel like the real power of it is in like a lot of the form sense of chemicals, because now you've collected these really complex, like super dense microbial cultures that now are full of all these different enzymes and cofactors and all these other things that now will go into the soil and feed the natural soil food web, and the microbes that are already there, and potentially turn them on and make them more active.
That's, from my understanding, is, a lot of these microbes are endemic in most soil systems. They're different categories in different places, but there's all these different functional systems that exist pretty much everywhere. And we've decimated through tillage or chemicals or lack of cover, but by using cover crops, which are exudates and all these different quorum sensing chemicals to feed microbes, I feel like it's a similar thing. You're taking this diverse microbial culture with a lot of quorum sensing chemicals and then dumping it into the soil, which wakes everybody else up and then causes a tremendous microbe.
We don't know a lot about the quorum sensing aspects. It's something that people are talking about more and more. Quorum sensing in the sense that, for example, when you looked at the root tip and you had that microbes there, you would anticipate, that's like a biofilm on the root tip of the root, right? That quorum sensing is going to play a role in that, right? And so the more microbes are there, maybe the more you have that will go in, right? That would be the idea. Same with the disease, for example. Quorum sensing in disease, you have to have biofilms. Essentially, quorum sensing has to happen. And once the biofilms are there, then the microbes can infect the animal, right? So that has to do with that. But there's also, it's a little more complicated in that there's also these other molecules that have the opposite effect, right? Quorum quenching and stuff like that.
Maybe quorum wasn't the rest, but signaling molecules.
Well, and we don't understand that. And we don't understand it, but it's likely, eventually we'll figure that out, right? We'll get a bunch of really smart people, physiologists, molecular biologists, whatever they are, right? And they're going to spend a lifetime on it, and they'll tell us. We'll learn from that. Yeah. We'll take our smartest people that we have, we'll put them on this. Yeah, that's what. And so we'll go past that barrier, right? We'll go past that barrier eventually. We'll get through that. Yeah. Any other last questions, or are we going to see some stuff now, or? Okay, this is nice. Does anyone recognize what this is? Where did Ruben go? Anyone recognize what this is? This is actually one of those mucor things. This is the sporangium of a mucor. And you can see it, you can see the sporangium here, and you look inside there, you can see little spores in there. You see some spores hanging on the outside too. This might be spores of something else. This might actually be mucor itself here. There's another one. This is another sporangium. You can see that it's a head and it's got full of spores in there. The stalk is missing on that one. There's some hyphae. Oh, that is cool.
Isn't that cool?
That is
that's
cool.
the coolest thing I've ever found in a microscope.
You know what it is, right?
Nematophagus.
Those are constriction rings. So the way these things work is a little ascomycete. The way they work is a nematode comes along, and swims through these rings. This fungus puts these out in a film of water. The nematode swims in the film of water, comes there, puts its head in there, and as soon as it goes into that ring squeezes down, catches that nematode. And then what happens is the hyphae, it may break this hyphae off, but the constriction hyphae will grow, and it'll grow through the skin of the nematode, and it will grow in the nematode and eat it out from inside out. These are really cool. That's
I also have a video, I think, of one that I went down a deep wormhole on these because they're so cool. It was
cool.
stuck on something. And it could have just been stuck on something else. I couldn't quite get it in, it wasn't in the right line of sight, but.
They have nets.
Yeah, there's ones that have like they
nets.
And they like mace things as they go by. Like, it's wild. It's so cool.
Well, of course, these fungi want nutrients too, right? So they catch a nematode and they got a big bag of nutrients right there. So they can eat it up, eat those things up.
I have no idea what that is.
Okay, yeah, well, we got debris in here. Probably, I think I see bacteria hanging around in here. Maybe some yeast in here too. This, now that, maybe it's an oospore. Oospore belongs to an oomycete. And looks like it, a thick wall. Could also be an egg of something, but maybe it's an oospore. Just a resistant oomycete spore. Could be a spore that belongs to something else too. It's kind of hard to say when we're looking at, just look at that.
You
Yeah.
mentioned about yeast being one of the fungi that's taken in the Rhizophagy cycle. Is there any indication of what function or what nutrient or what that provides and why that it's mostly bacterium rather than
There is not. And it should be harder to get nutrients out of yeasts. The only thing is that many of those yeasts carry bacteria inside them. So they may be getting some, breaking some of those up, getting the bacteria out of those yeasts. We don't know a lot about it, right? So there's still a lot that has to be learned. Okay, so here I see something in here. You can see I see a little loop-like thing. I think this could be a nematode egg. Not totally sure, but I think there may be a nematode in here. This is some kind of an egg, though. I'm pretty sure of that. It's big, too.
It's huge.
It's huge, yeah. I think it's a nematode egg. Nematodes lay these eggs, and they lay a lot of eggs.
So interestingly enough, this is from IMO 3, which one of the kind of contentions of natural farming is that it shouldn't have nematodes in it. But often does, so.
Well, I mean, we'd have to confirm it. I want to see what this.
Hatch the baby.
If it hatches, did you see
Find
it?
this egg.
We'd have to hatch
Pop it in
it.
a bag, I know where it is.
Or when it develops more, you may be able to see the nematode curled up there in there better when it develops more. Yeah, if that truly is a nematode. Yeah, right?
That was the whole thing.
Yeah, yeah.